Energy-converting organelles. Chloroplasts reside in plant cells and some protists and convert solar radiation into energy stored in the cell for later use. Mitochondria reside in all eukaryotic cells and convert chemical energy from glucose into ATP. Interestingly, both chloroplasts and mitochondria have their own DNA, separate from that found in the nucleus of the cell. They also have a double cell membrane. These organelles convert solar energy to chemical energy through photosynthesis. Chloroplasts are partitioned into three major compartments by internal membranes: thylakoids, stroma, and intermembrane space. Remember that during photosynthesis, it is on the thylakoid that the light-dependent reactions take place. In prokaryotes, thylakoids are areas of highly folded membranes. In eukaryotes, they are stacked in the chloroplasts. The fluid outside these stacks of thylakoids is called the stroma. This is where the Calvin cycle takes place. Mitochondria are sometimes referred to as the powerhouses of the cell. They convert chemical energy, or glucose, into a more usable and regenerative form of chemical energy, ATP. The mitochondria is also partitioned like a chloroplast. They only have two compartments as opposed to three in the chloroplast, the matrix and the intermembrane space. Remember, cell respiration must take place near a membrane so that a proton gradient can be built in a membrane space that is separate from the rest of the cell. Thus, the membrane would separate the inner volume with a deficit of protons from the outside with an excess. In prokaryotes, the intermembrane space is between the cell membrane and the cell wall. In eukaryotes, that membrane is the intermembrane space of the mitochondria in between the inner membrane and outer membrane. The mitochondria and chloroplast are different from other eukaryotic organelles because they have their own DNA, their own ribosomes, and have a double cell membrane. In 1970, Lynn Margulis published The Theory of Endosymbiosis to explain these facts. The theory states that the mitochondria and chloroplast were once free-living prokaryotes that got taken up or eaten by another prokaryote. The mitochondria was a bacteria that could make its own ATP. The chloroplast was a bacteria that could perform photosynthesis. When they got taken up by another prokaryote, they dragged the one prokaryote's cell membrane around theirs, thus the double cell membrane. This now allowed the new eukaryote to make its own ATP or be able to do photosynthesis and make its own food, thus the evolution of eukaryotes. Note, the nucleus and flagella could also have the same roots, although they are not as heavily supported with evidence as the mitochondria and chloroplast. Both mitochondria and chloroplasts can arise only from pre-existing mitochondria and chloroplasts. They cannot be formed in a cell that lacks them. Both mitochondria and chloroplasts have their own DNA, and it resembles the DNA of bacteria, not the DNA found in the nucleus. Both mitochondrial and chloroplast genomes consist of a single circular molecule of DNA, just like in prokaryotes. Both mitochondria and chloroplasts have their own protein synthesizing machinery, and it more closely resembles that of bacteria than that found in the cytoplasm of eukaryotes. Since mitochondrial DNA is not in the cell nucleus, it is only passed along from mother to child. Animals, including you, inherit your mitochondria from your mother only. This is because the egg from our mothers contained her organelles. Dad's sperm only contains the chromosomes, none of his organelles usually. All of our organelles we inherited from our mothers. Mitochondrial DNA is a way to trace maternal heritage through a family or through a species. The mitochondrial Eve is the first human female that gave rise to all humans. In theory, we can trace all humans back to her through our mitochondrial DNA.